This brief lecture covers evolution of pathogens, and the basis for evolution, obviously, is genetic variability. And one of the most fundamental aspects of genetic variability are random mutations, usually point mutations, where individual bases are exchanged for the wrong base. And so the prototypical system for looking at high rates of random mutations are the RNA viruses, because they have super high mutation rates, typically due to the fact that they have low fidelity polymerases, meaning that their polymerases are error prone. They don't have error correcting mechanisms like ours do, for example. And as a result, when they make a mistake, they can't go back and remove the mistake and add the correct base. And so these mutations build up. And that when you couple that with their high replication rate, they not only build up, they magnify incredibly over time. Now, obviously, that results in a lot of extinctions. So a lot of the progeny go extinct because they have too many mutations or they have key mutations in terrible places. But because there's so many of them, natural selection is acting intensely on the few that are very successful. And those few that are very successful can then spread. And so HIV's reverse transcriptase is a good example of a low fidelity polymerase. It makes errors in about 1 in every 30,000 bases that it lays down. But when you're a virus that's making billions of progeny every day, that means that at this rate, there's going to be mutations in just about every gene in the genome by the time the virus progeny start to break out of the cell. And as a result, RNA virus populations are really considered quasi-species, or a mutant horde, as we like to call it, meaning that you have a single type of parent that enters the cell, and then what comes out of it is, again, just a whole horde of different mutants, and with selection acting on those mutants and selecting for the most successful, the most transmissible, and so forth. Another version of genetic variability that occurs in RNA viruses is recombination. And so in this particular case, you can imagine 2 RNA viruses that have infected the same cell. And then that sets up a situation like this, where you have a donor and an acceptor. The donor in this particular case is just whatever virus genome that got off sooner. And as a result, its polymerase has already started replicating that particular genome. But in this particular case, the polymerase grabs the 5' end here and jumps ship and grabs onto a hunk of RNA from a genome of a virus that was nearby. And as a result, it completes replication on that acceptor, what's called the acceptor genome. And so you end up with this hybrid genome. So part of this genome, as you see here, came from the donor, and then the other part of this came from this acceptor. And so this obviously is a much more intense version of genetic variability than if this were just this complete genome here, but with a few point mutations. A version of recombination that occurs often in DNA viruses, but also in bacteria, which are DNA-based, is illustrated here. So this is an example of homologous recombination, where you have imagine this is a genome, a double-stranded DNA genome, and then a microbe that takes in this hunk of DNA, this hunk of double-stranded DNA through any number of mechanisms, which I'll show you. If there is homology between this fragment here and this fragment here, then these two will cross over, just like crossing over that happens in meiosis, for example. This crossing over event leads to homologous recombination. So again, these guys are homologous, and these guys are, and as a result, hydrogen bonds formed, and then ligases and enzymes will break the phosphodiester backbones and seal these up, and so you end up with these homologously recombined unique genomes. And so then, in subsequent rounds of replication, this strand, when it replicates discontinuously, this strand will have its opposite strand will have something that more closely represents this. You got to remember that doesn't have to be 100% match here. So it's just close enough to keep it stitched together long enough so that when this is replicated, the daughter strand that comes from this will be very closely aligned to it, 100% aligned to it. Phenotypic mixing is another thing that happens in viruses, but this is not a genetic alteration. This is a physical protein alteration. And so here's a situation where you've got two different viruses that co-infect the cell. And as these guys are replicating, as this genome and this genome are replicating and making virus pieces, these pieces get shuffled in the inside the cell. So even though this guy got the same genome as its parent, and this guy got the same genome as its parent, it's assembled with bits of this virus, and this guy, and this progeny virus is assembled with bits of this guy. And so there's a combination. So there's no change in the genome, but one possibility is that there's an altered host range. So now, if this guy is more as an influenza virus, for example, that's more adapted to infecting geese, and this is an influenza virus that's more adapted to infecting pigs, now you've got these two hybrids that should be relatively equally good at infecting both dogs and pigs, or geese and pigs, or whatever. And so another interesting aspect of it is could become possibly resistant to antibody neutralization. If your body produces antibodies against just this particular set of spikes, and now the progeny have a combination of spikes, any neutralizing antibody that binds to this will no longer block this. And so this virus still may have the capacity to bind to a host cell and infect. A really cool example of genetic variation is reassortment that happens in influenza viruses. In this particular case, the same type of scenario happens in which two influenza viruses, infect the same animal, and in fact infect the same cell of the same animal. And so in this particular case, because these viruses, as you see here, have genomes that come in segments, and typically 7 to 8 pieces, that allows them to exchange entire RNA molecules between genetically related viruses that also have segmented genomes. So the virus 1 and virus 2 both infect this cell, and their genomes are replicated into pools, and then there's some mixing that can happen. And then the assembly of new virus particles results in a variety of outcomes. One outcome might be, this virus is the same as the original, and this virus right here is the same as the other original. But often there's a giant pool of these new variant viruses against which there's no protection. So now this guy is an amalgam of these two. Genetically, it's an amalgam. Now again, here, the genomes are the same. It's just the pieces that are different that make up the virus. And here, the genomes are different, and as a result, their spikes will eventually be slightly different from the new genome bits that they have. Horizontal gene transfer can happen usually through these three mechanisms. The first over here is indicated is conjugation, in which the two bacteria, for example, come together through a mating pilus, and then that mating pilus allows for the donor that you see here to give up its plasmid to a recipient. Another version of horizontal gene transfer is pictured here, transformation, where this cell has lysed for some reason. It could be chemicals or osmotic stress, and bits of its genome have broken up and gone into the environment. And then some of these pieces enter the cell and then can homologously recombine into the genome. And again, you can imagine it looking something like this at that interface between the incoming DNA and the genome. Another example is transduction, in which genes are passed through viruses. So this bacterium, for example, is infected with a virus, but the virus, rather than packaging its own genetic information, will package bits of this host's DNA, and then the virus goes out into the world and injects that host's DNA into this bacterium. So that would be transduction if the gene is stably incorporated, or if the DNA is stably incorporated and gives this microbe novel characteristics. So here's an example of the medical significance of horizontal gene transfer, and here's a medical significance of transduction in particular. So this Shigella bacterium, that has this deadly Shiga toxin gene, is infected by this phage. And then this phage destroys this cell, but rather than package its own genetic information. It cuts up the host DNA and packages bits of the host DNA instead. So this thing only becomes basically a carrier vector for this guy's DNA. So this cell's DNA then is injected into a recipient. In this particular case, it's a mild diarrhea-causing E. coli. So this Shigella is a severe bloody diarrhea-causing Shigella, and its bloody diarrhea-causing DNA that you see here is transferred to this mild diarrhea-causing E. coli. If that DNA can incorporate, as you see here, you suddenly have a new strain of E. coli that's capable of causing bloody diarrhea and kidney failure and all those types of things. This over here is an example of either transformation or mating or conjugation. So in either case, the bacterium, the donor that you see here, can either mate with another strain, or will be lysed somehow and give up its DNA to this guy in that way through free DNA uptake. The net result is some kind of homologous recombination event of that new DNA into the genome. So you've got a penicillin-resistant strain and the penicillin-susceptible recipient. And as a result, this penicillin-susceptible recipient takes in that DNA and becomes resistant. The interesting aspect of this, another interesting aspect of this evolution is in bacteria, is this process of evolution and leaps and bounds through pathogenicity islands. And so in this particular case, microbes can acquire huge chunks of DNA. And so in these examples here, it's a resistance gene or a toxin gene, but often these are huge blocks of genes that can be acquired, and giving this microbe not just a single trait, but a whole suite of traits. So the microbe can gain resistance to a whole bunch of antibiotics, or it can gain an operon necessary to eat a novel carbon source. Or it could even change its respiration details so that the microbe can suddenly become aerobic. But so that's what's pictured here, is this concept of islands of DNA. And if the islands of DNA happen to bestow on the microbe pathogenicity traits, then we call them pathogenicity islands. And so these are transmissible type DNAs that often are associated with transposons. So again, transposons being jumping genes that allow hunks of DNA to enter genomes or cut themselves out of genomes and move into other bacteria through any of those three horizontal gene transfer mechanisms that I mentioned. An example of that is the presence of pathogenicity islands that correlates with pathogenicity, such as these pathogenicity islands SPI1 and SPI2 in Salmonella. And I'll talk about these, but or I have talked about these already, that SPI1 and SPI2 are the Salmonella pathogenicity islands that encode the various type 3 secretion systems that the bacteria use to get inside cells and survive intracellularly. A single pathogenicity associated island can transform a benign microbe into a virulent pathogen. So if a microbe gets a whole bunch of genes for antibiotic resistance, or a whole bunch of genes for type 3 secretion or toxin production or whatever, the microbe can transform and automatically become, through one evolutionary event, they don't have to wait millions of years for it, they can become deadly pathogens, for example. Chromosomal pathogenicity islands, as I've already mentioned, are often inserted into the genome, and usually they're inserted at very specific regions of the DNA, so at 3' end of tRNA. And this is something that phage often do. When phage insert themselves, they look for various sequences that often occur at the 3' end of transfer RNA genes. And so the fact that PAIs do that as well suggests that they may have originated from phage, from bacterial viruses. Some of the things that we look for in terms of evidence for recent horizontal transmission is the GNC content of DNA. So you can analyze DNA, and if you see a microbe with an unusual set of genes, you can count the G's and C's that make up that block of DNA and compare that to the rest of the DNA in the genome. And the more ancestral DNA should smooth out essentially and have a more consistent GNC content. But if you find a big block of DNA that has, 40% GNC content, and whereas the rest of the genome has 25% GNC content, you can tell that probably more recently came into that microbe. Another aspect is codon usage, the triplet code that the microbes might use to encode individual amino acids changes as well. And so sometimes microbes over time will start to favor a particular set of codons to encode particular amino acids. But if the DNA has recently come into the cell, they may have different codon usage. So they may use different triplet codes to encode amino acids that the rest of the genome isn't using. And that's an interesting way to look for horizontal gene transfer. Often these PAIs are flanked by repeats, DNA that occurs in repeating sequences. That suggests that it's an insertion site. And the other thing that we look for is the presence of phage genes. So are there capsids and tail fiber proteins and stuff encoding genes around the insert? And that, again, would give us the sense that there is a possible insertion of novel DNA or new DNA of phage origin.